Welcome to this lecture on the histology and regulation of the gonads. Today, we're going to explore one of the most fascinating and vital systems in the human body. The gonads are the biological foundation of human existence, responsible for both creating life and maintaining our overall systemic health. We will start by building a strong, intuitive understanding of how these organs function. Then dive into their microscopic anatomy, and finally, we will use mathematical models to see exactly how the brain controls them. By the end of this session, you will have a comprehensive understanding of both the biology and the quantitative dynamics of human reproduction. Let us begin our journey into the endocrine system. To start, let us imagine the gonads as a highly advanced manufacturing plant that has two very distinct but equally important jobs to perform every single day. Why does this matter? Because understanding this dual role is the key to understanding all reproductive biology. First, this factory needs to physically build a complex product, which in our body's case represents the creation of the sperm in males or the egg in females. Second, it needs to broadcast chemical signals to the rest of the city to let everyone know the factory is running smoothly. This represents the secretion of sex hormones like testosterone and estrogen. The gonads, which are the testes and ovaries, do not just work on their own. They are constantly receiving orders from the chief executive officer in the brain, specifically the hypothalamus and pituitary gland. Now that we understand the dual factory analogy, let us look at the actual microscopic rooms inside the male factory. In precise medical terms, the gonads are responsible for gametogenesis, the production of gametes, and steroidogenesis, the production of steroid hormones. Histologically, the male testes are divided into two main compartments. First, we have the seminiferous tubules. Inside these tubules, you will find nurse-like Sertoli cells. Their primary job is to nurture and support the development of sperm cells. Outside of these tubules is the interstitial space. This area contains the Leydig cells, which are the heavy lifters for hormone production, specifically manufacturing testosterone. Understanding this spatial separation is crucial because it explains how the testes can simultaneously manage physical sperm production and chemical hormone synthesis without interference. Moving from the male anatomy, let us examine how the female factory is structured. In females, the ovaries operate a bit differently. Instead of tubules, the ovaries contain developing follicles. You can think of a follicle as a functional, self-contained unit. At the center of this unit is the oocyte, or the developing egg. Surrounding this precious oocyte are two vital layers of supporting cells, the theca cells on the outside and the granulosa cells on the inside. Just like the Leydig and Sertoli cells in males, these female cells must cooperate perfectly. The theca and granulosa cells work together in a complex chemical relay to produce estrogens. This microscopic teamwork is what allows the ovary to mature an egg while simultaneously preparing the rest of the body for potential pregnancy through hormonal signaling. Having seen the cellular workers, we must now understand how the brain manages them. The regulation of these microscopic structures is strictly governed by the hypothalamic pituitary gonadal axis, often abbreviated as the HPG axis. This is the chain of command. It starts at the top with the hypothalamus, which secretes gonadotropin-releasing hormone. This hormone acts as a memo to the middle manager, the anterior pituitary gland. In response, the pituitary releases two critical messengers, luteinizing hormone and follicle-stimulating hormone. These hormones then travel through the bloodstream all the way down to the gonads. Once there, They bind to specific receptors on the Leydig, Sertoli, theca, and granulosa cells we just discussed, giving them the direct order to start producing gametes and steroids. Before we dive into the mathematics of this signaling, let us discuss why this delicate axis is so clinically important. Understanding the histology and regulation of the gonads forms the biological foundation of overall systemic health. Without precise coordination between the brain and these specific gonadal cells, reproduction would be entirely impossible, leading to permanent infertility. Furthermore, the hormones produced by these tissues do much more than just regulate reproduction. Estrogen and testosterone actively maintain your bone density, muscle mass, cardiovascular health, and even your emotional mood. When this delicate regulatory axis breaks down, patients can suffer from severe, life-altering conditions like polycystic ovary syndrome, hypogonadism, or hormone-dependent cancers. Mastering this topic is what allows doctors to diagnose and treat a massive variety of endocrine disorders effectively. Now that we appreciate the clinical stakes, let us zoom in to the molecular level to see how these signals are actually received. Hormones like LH and FSH are completely useless floating in the blood unless they successfully bind to specific protein receptors located on the surface of target cells, such as the Leydig or granulosa cells. Think of the hormone as a key and the receptor as a lock. The strength of the biological response generated inside the gonad depends entirely on how many of these locks are successfully turned by the keys. To truly understand and predict how strongly a gonad will respond to a specific hormonal signal sent from the pituitary gland, endocrinologists rely on a specific mathematical model, which we will look at next. This brings us to the receptor occupancy equation, which mathematically describes the relationship between hormone concentration and activated receptors. Conceptually, this formula comes from the basic principles of chemical equilibrium, balancing the rate at which hormones bind to receptors against the rate at which they fall off. Let us break down the symbols. B represents the number of bound receptors, which dictates the strength of the cell's response. B max stands for the total maximum number of receptors physically available on the cell surface. The bracketed H represents the concentration of the hormone currently circulating in the blood. Finally, Kd is the dissociation constant. It measures the affinity of the receptor for the hormone. A lower Kd means the receptor binds the hormone very tightly. If hormone concentration increases, bound receptors increase. If Bmax decreases due to disease, the maximum possible response is severely limited. Let us apply this formula with a concrete clinical example. Imagine we are evaluating a male patient to see how his Leydig cells are responding to luteinizing hormone. We are given that his circulating hormone concentration is 2.0 nanomolar. The dissociation constant is 0.5 nanomolar, and the maximum number of receptors per cell is 10,000. We need to calculate exactly how many receptors are currently bound. First, we substitute our known values into the receptor occupancy equation. The numerator becomes 10,000 times 2.0, representing the theoretical maximum binding capacity multiplied by available hormone. The denominator becomes 0.5 plus 2.0. Simplifying this, we get 20,000 divided by 2.5. Performing the final division, we find that exactly 8,000 receptors are bound. This means 80% of the receptors are active, indicating the patient's gonads are receiving a robust stimulatory signal to produce testosterone. Now that we have seen how hormones initiate a signal, we must understand how that signal is turned off. Gonadal regulation is highly dependent on the body's ability to clear hormones from the blood. Biological signals cannot last forever. If they did, the system would be permanently stuck in one state. The liver and kidneys continuously remove these hormones from the bloodstream. This metabolic clearance typically follows what we call first-order kinetics, meaning the rate of clearance is proportional to the amount of hormone present. Understanding this clearance is absolutely crucial because as the hormone levels drop, the negative feedback on the hypothalamus is finally lifted. This lifting of suppression is exactly what allows a brand new reproductive cycle to begin. To predict exactly when that negative feedback will be lifted, we use the standard exponential decay formula. Conceptually, this mathematical model represents the continuous, compounding removal of molecules by the body's metabolic organs over time. Let us define the variables. C of T represents the concentration of the hormone remaining in the blood at any given future time t. C0 represents the initial peak concentration of the hormone, right after it was secreted by the gonad. The mathematical constant E is the base of the natural logarithm, used to model continuous decay. The variable K is the decay constant, representing the specific rate of clearance by the organs, and T is the elapsed time. As time increases, the negative exponential term becomes progressively smaller, which mathematically ensures that the hormone concentration steadily decreases over time. Let us see this exponential decay in action. Suppose a female patient's ovary produces a sudden burst of estradiol, reaching an initial peak concentration of 400 picograms per milliliter. The decay constant is 0.05 per hour, and we want to find the concentration after 10 hours. First, we plug our known physiological values into the model. We multiply the negative decay constant, 0.05, by the time, 10 hours, to get a simplified exponent of negative 0.5. Evaluating e to the power of negative 0.5 gives us approximately 0.6065. This means roughly 60% of the initial burst has survived clearance. Finally, multiplying this decimal by the initial 400 gives us 242.6 picograms per milliliter. This remaining concentration is likely still high enough to exert negative feedback on the pituitary gland. Having calculated the dynamics, let us bring everything together visually in this diagram of the HPG axis. At the very top, you see the hypothalamus, which initiates the cascade by sending GnRH down to the anterior pituitary. Notice the two diverging arrows leaving the pituitary, LH and FSH. Follow the LH arrow, and you see it targets the interstitial Leydig cells and theca cells, driving them to produce testosterone and estrogen. Follow the FSH arrow, and it targets the nurse-like Sertoli and granulosa cells to support gamete development. Crucially, look at the long arrows looping back up to the brain from testosterone and estrogen. These are marked with minus signs, representing the negative feedback loop we just discussed. This diagram beautifully summarizes how structural histology and chemical signaling unite to regulate human reproduction. We have covered a tremendous amount of ground today, so let us reinforce our key takeaways. First, remember that the gonads are complex, dual-function organs. Whether it is the Leydig and Sertoli cells in males or the theca and granulosa cells in females, they work together to produce both gametes and steroid hormones. Second, this entire system is masterfully controlled by the hypothalamic pituitary gonadal axis. Third, we learned that this regulation is not just qualitative, but quantitative. Receptor binding kinetics dictate exactly how strongly a cell responds, and exponential hormone clearance dictates exactly when the system resets. By mastering both the microscopic biology and the mathematical dynamics, you now possess a holistic understanding of human reproductive endocrinology. Excellent work today, and thank you for your attention.